you have to remember, not a compulsory, but it gives you an idea like how you can remember your the different types of substrates and products and your enzymes. See this, whatever year can I keep selling substance for money, officer, that is your substrates what are involved in your TCA cycle. Okay, citrate, isocitrate to fumarate, malate and oxaloacetate. So this side, whatever I have put, always remember the main enzyme involved in your TCA cycle is dehydrogenase. Dehydrogenase is the main enzyme involved, and S stands for synthase. So keeping in this in mind, so at disco, devil sit down 5 drinks. So you have it. Whenever S come, blindly write it as synthase. Then citrate synthase. Acortinase you have to remember. So then isocitrate dehydrogenase, again dehydrogenase, S means synthase. Here again you have to remember F means fumarase. Okay, and then you remember that even FAD is formed. Here succinyl CoA to succinic acid, remember that GTP is formed, and F I told before that FADH2 is formed. So these are the tricks that you have to remember. You can keep it in your mind and remember. Now quickly we can go for our TCA cycle. Very simple regulation cycle in TCA. As I told you, why regulation is important. Regulation means, in simple step I told you, it is switching on and off like a switch it is. So what did we tell, how we defined regulation? We defined it as telling that, like, in order to maintain, okay, so many metabolic activities are going on in our body. In order to maintain equilibrium or stability or homeostatic condition, okay, our body regulates the metabolism. Is a particular cycle should go on, or should I switch it off? That is what is your regulation means. And this is brought about by certain substrates or your products or cofactors from your body, from that particular metabolism itself. So when you regulate, what happens? Your body is in equilibrium, it is in a stability. So what is the outcome? That I can interact, okay, I can respond to the external stimuli of my environment. That is what in regulation means. If it is a reaction is required, you proceed with the reaction. If it is not required, do not proceed it. Do not waste your ATP. That is what our cells or our signals are telling our body. Now, completely, I'm not telling that you stop your reaction. That is slow down your reaction. Otherwise, speed up your reaction. This is what we mean by regulation. It is just controlling. Now, when you come to the TCA cycle controlling, you can see here all the reds which are marked like this, okay? That is all is your inhibitor. Whereas the green ones are your activator. So the first one we shall take and finished it off. Here we have what? Pyruvate is getting converted to acetyl-CoA. So acetyl-CoA is your first substrate which is formed in the TCA cycle. And what is the enzyme involved? Pyruvate dehydrogenase complex. Look at this. Your ATP, acetyl-CoA, NADH, fatty acids, all are inhibitors of your pyruvate dehydrogenase. It will not allow the reaction to proceed further. Now you know the answer why. Because I told you, whenever there is ATP, why ATP is inhibiting a reaction? It is inhibiting a reaction. It is indicating that high energy is almost present in the body. We don't require anymore. I am already sufficient amount is present. Why are you going to produce me again? So it will inhibit your pyruvate dehydrogenase. So also acetyl-CoA. See, acetyl-CoA is what? It is a product of the pyruvate. So it will send a negative feedback telling the pyruvate dehydrogenase acetyl-CoA is a product. It can induce negative feedback. That is, it is telling that there is an accumulation of acetyl-CoA. I am getting accumulated in the TCA cycle. What does this mean? It means that I am in lot, okay, I am more in number, and I'm not getting converted into citrate or further on. It means to tell that my TCA cycle is already backup I'm having. So there is no need for me to get converted into citrate. So if you're going to produce me, I'll just get accumulated. So it is just a waste. So you stop the reaction. That is what your acetyl-CoA is telling. Similarly, you have your NADH. Again, we know about the NADH. So it is again inhibiting who? It is inhibiting your pyruvate dehydrogenase. That is whenever the NADH concentration is high, okay? Whenever the NADH concentration is high, it is a product, okay? See here all NADH is being formed here. What is it? It is a product which is formed. It is indicating that more of NADH is being produced and it is not getting reoxidized to NAD. So I have sufficient amount. TCA cycle is telling that I have sufficient amount of NADH. That is why the concentration is more. So I'm not getting reoxidized to NAD. So please stop the reaction because we are in sufficient amount. So fatty acid. How your fatty acid is inhibiting pyruvate dehydrogenase? Fatty acid mediated inactivation of pyruvate suggests that addition of long chain fatty acid to the tissue may cause increase in ATP-ADP ratio. So it is telling, see, if my concentration is already high and if you're going to proceed with the reaction, I'm going to again start producing long chain of fatty acids. So it indicates what? That again there is an increase in ATP. If there is increase in ATP, so much of ATP is produced. So what is the point of it? It just goes waste. So what it is telling is pyruvate, please don't continue with the reaction. We are also in sufficient amount. So these all act increase in as ATP, NADH, fatty acid, acetyl-CoA, all are inhibitors telling that we have sufficient backup with us. So you need not speed up the reaction. You can go slowly or you can stop the reaction. Similarly, here also you can see NADH, succinyl-CoA, citrate, ATP. We'll not study about NADH and ATP because we have already spoken up. Now, what is the succinyl-CoA? Why it is inhibiting? And why? What about citrate? Both again same thing they're telling. The succinyl-CoA and citrate are telling to the reaction system. They're telling what they're telling. We are accumulated and it is not getting converted. Okay. See your succinyl-CoA. Where is your succinyl-CoA? Here it is. Succinyl-CoA. Where is your citrate? Here it is. So it is telling that particular enzyme we are getting accumulated and we are not getting converted. Citrate is not getting converted to isocitrate. Succinyl-CoA is not getting converted to succinate. Okay. So if it is not getting converted, what it means? I become accumulated. I am increased in concentration. So what it indicates is all others are sufficient in my TCA cycle. Simply why you want to continue with the cycle when we are in sufficient amount and other reactants don't want us because we have again a backup of a reaction. That is the meaning of succinyl-CoA and citrate. We are already present in sufficient amount. Always whenever there is an inhibitor, kindly remember they are telling that switch off the reaction. To whom they are telling? To the enzyme they are telling. What are enzymes? They are, we told they are proteins. And what are proteins? What is making a protein? It is the DNA. What is your DNA? It is the gene. So they are switching off the gene. Don't produce it anymore or produce less amount of it because we are always having some backup with us. We are not insufficient of the material. So look at this green color. What we told, whenever there is green, there is a green color, it indicates that it is an activator. It is telling your enzymes. In turn, it is telling your proteins. Proteins is in turn telling the genes, come on, we are less in amount. Start speeding up the reaction. See, whether it is ADP or AMP, there is no ATP. So it is telling, please enzymes carry out your reaction. We don't have sufficient amount of energy in a cycle, so speed up the reaction. That is what your AMP, CoA, NAD, ADP all are telling. You activate the reaction. Here you can see even calcium is acting as an activator. What is the function of the calcium? Calcium activates so that more ATP is produced in your muscles. Because I'm feeling my muscles are feeling tired, it indicates that, okay, I don't have energy. What it means when I don't have energy means there is less amount of ATP is being produced. So calcium immediately it signals your TCA cycle telling that muscles are not having energy. There is no ATP. Please proceed with the reactions. So this is what your activators and inhibitors are doing. This is what you mean by regulation. Regulation means simply switching on and off the gene. If I want, you proceed it. If I don't want, you stop or slow down the reaction. That is the meaning of the regulation. So here you can see even NAD. Yeah, here NAD, even your CoA, they are also activators. So NAD is an what it is telling that you activate. So more NAD are waiting to get reduced into NADH. Okay, I am more in number. So what you have to do, you activate the reaction. Only then I can get converted into NADH. Otherwise, simply I'll be getting accumulated. What is the use of me getting accumulated? I have to get converted into NADH. I have to get converted into FADH2. Only then these two can go into your TCA cycle and produce ATP. So they are all activators. In simple terms, students, remember whenever a point comes, regulation means in simple word, you have to remember that it is switching on and off your gene. Okay, so that your body is in the equilibrium, or the word what we use is the homeostatic condition. Only when we are in equilibrium, only then we can react to the external stimuli. We can activate towards the external stimuli. So metabolism is going on in your body. So if there is an accumulation of some products, and if it is not necessary, then already your body have a mechanism to tell that don't continue with the reaction. Don't waste anything. Or if there is shortage of any product or your energy, then it sends another signal, the reverse signal telling that we are less in amount. You have to produce, you have to continue with the cycle. Only then I can sustain. So finally, the bioenergetics. This is important. In your TCA cycle, what was your product? Reactant acetyl-CoA, 3-NAD, FAD, GTP. So I'll show here only. See, this all. I got acetyl-CoA. Then what was that? NAD was present here. FAD is present here. GDP is present here. That is what we have written here. And what happens? They are getting converted to 2 molecules of carbon dioxide, 3 molecules of NADH, and 1 molecule of FADH2 plus 1 molecule of GTP. This is the end product of my TCA cycle. So I told you that GTP is nothing but an ATP. The same function of ATP only it will have. Only it is a gonosine. And what did I tell you about FADH2? Whenever FADH2 is produced or NADH is produced, they go to the ETC cycle. When they go into the ETC cycle, what did we tell? They will get converted to ATP. So always one molecule of NADH will get converted to 3 molecules of ATP. Okay. So again, 1 molecule of your FADH, remember 1 I am speaking, 1 molecule of FADH will always get converted to 2 molecules of ATP. So that is why we told that as TCA cycle, 1 acetyl-CoA is going to give us 12 molecules of ATP. And we know that there are two molecules of pyruvate, and therefore both the molecules is going to produce two molecules of acetyl CoA. So what happens? I have got 12 molecules of ATP. Again, if I cut two times, if the cycle is going on, I'll be ending up in 24 molecules of ATP. This is the bioenergetics which you have to remember.